Boa noite a todos. É com enorme prazer que nós voltamos ao nosso simpósio. Eu tenho algumas coisas para colocar antes de dar a palavra aos acadêmicos. Antes, eu tenho que cumprir o ritual aqui, que é declarar aberta a 24ª sessão ordinária relativa ao 196º ano acadêmico, e a gente precisa aprovar a ata da 18ª sessão ordinária do dia 31 de julho de 2025. Alguma observação? Ok. Bom, a primeira coisa é eu queria dar os parabéns efusivos ao nosso acadêmico Alexandre Siciliano, que está entre os melhores cirurgiões de coração nos Estados Unidos, reconhecido pelos Estados Unidos entre os 17% melhores cirurgiões. De modo que eu gostaria de parabenizar efusivamente o acadêmico siciliano. Infelizmente, ele não está aqui, mas eu, mais tarde, vou ligar para ele para dar os parabéns. A outra coisa, acho que ele merece mesmo. A outra coisa é que a nossa reunião com a National Academy of Medicine dos Estados Unidos está dando bons frutos e nós estamos tendo o aceite de participar na nossa manifestação sobre clima e saúde de um número muito grande de academias nacionais. Nós estamos agora perto de 50 academias nacionais de medicina que aceitaram participar da reunião, de ter um manifesto, não sei se irão à COP30, mas de ter um manifesto para a COP30. Conversando hoje com o presidente Belfort, nós pensamos também em fazer uma reunião conjunta com a Academia Brasileira de Ciências, e eu acho que um possível tema pode ser também clima e saúde. Então, eu vou ver quais são as datas que nós temos disponíveis para propor à presidente Helena Nader, com a intermediação do nosso presidente Rubem Belfort, para organizarmos um simpósio conjunto entre as duas academias, porque nós temos aqui também membros da Academia Brasileira de Ciências, inclusive da seção Saúde, e o Marcelo Barcinski, que você não é da seção Saúde, é? Acho que você é da seção Biomédica. Você também é biomédico. Uhum. Eu gostaria de lembrar também que a World Academy of Science vai se reunir aqui no Rio a partir do dia 28 de setembro, na Barra. Eu devo comparecer, eu também sou membro da Atuais, e eu gostaria de convidar a todos para participarem da reunião. O que eu tinha que falar é isso, e a palavra está aberta para os acadêmicos. Acadêmico Rubem Belfort. Presidente Rubem Belfort.
Muito obrigado. Eu queria contar aqui, provavelmente vários já sabem, mas é importante. A nossa presidente fez uma conferência excelente na Academia de Medicina de São Paulo, com a presença de vários acadêmicos nossos ali. E, mais uma vez, ficou claro, não apenas o brilhantismo da presidente, mas a oportunidade da palestra dela, mostrando a necessidade que a sociedade brasileira tem de continuar contando com instituições como a nossa Academia Nacional de Medicina. Então, esse aumento da visibilidade da nossa academia e a integração com outras entidades, que eu também considero fundamental, tem sido muitíssimo bem estabelecido pela nossa presidente. Então, eu queria aproveitar a oportunidade para que todos soubessem disso, e eu, pessoalmente, gostaria de felicitá-la. Foi muito bom esse tipo de depoimento. Também queria lembrar do lançamento do livro, né? Professor Silvano Raia. Por favor. Não, presidente, por favor.
O acadêmico Silvano Raia lançou um livro que foi... Eu já comecei a ler, é muito interessante, aos 95 anos. Além dele ser um ícone da medicina, é um exemplo para todos nós, porque chegar aos 95 anos como o professor Silvano Raia realmente é uma glória. E ele fez uma coisa tão cuidadosa, porque o acadêmico Nardi, talvez você possa registrar o que você testemunhou. Você pode falar um minuto, por favor, acadêmico?
Senhora presidente, boa noite. Sim, foi um momento muito emocionante para todos nós, esse lançamento do livro do acadêmico Silvano Raia, e eu notei que tinha uma secretária que fazia dedicatórias no livro e ele assinava. Mas eu observei com outros acadêmicos e convidados presentes que cada dedicatória era muito personalizada, como se a secretária soubesse a história de cada um. E eu notei que ele já tinha feito as dedicatórias em um livro separadamente dos convidados, ela só copiava e ele checava novamente se estava dando o livro para a pessoa certa. Então, foi algo muito bem bolado por ele, muito bem articulado, e isso fez com que a noite e as dedicatórias fossem fantásticas. Obrigado.
Para terminar, e ele continua jovial como sempre. Hoje, no começo da tarde, por coincidência, o presidente Eliete e eu, que tinha vendido 164 livros, geralmente são 49.
Inclusive, ele estava tão bem que eu pedi a ele, eu tive um estudante, porque já defendeu a tese, que é transplantador de fígado, e o Silvano é o ícone, que ele gosta imenso do Silvano. Aí eu cheguei para o Silvano e pedi para ele que dedicasse o outro livro que eu tinha comprado. E ele sabia, a moça tinha uma dedicatória, porque eu tinha avisado que eu iria pedir esse livro. Quer dizer, ele estava totalmente organizado, gente, aos 95 anos. Realmente incrível, maravilhoso.
O que não tem, senhora presidente, nada de novo, né? Quem convive com ele, como eu, há 45 anos, cada vez que tem uma reunião, ele põe todos os itens da reunião que ele quer falar, é tudo anotadinho direitinho. E às vezes ele distribui para as pessoas para dizer que ele vai falar sobre aqueles. É impressionante como ele prepara bem cada reunião.
Chapô. Mas mais alguém gostaria de falar? Acadêmico Maurício Magalhães, por favor.
Excelentíssima presidente Rubens Belfort, Kalil, eu também queria fazer primeiro um comentário. Eu tive a oportunidade de estar no lançamento do livro e ler o livro. Eu li o livro esse final de semana, não consegui parar de ler, liguei para ele, fiquei uma meia hora conversando com ele. E é bonito que ele cita a academia muitas vezes, muitos acadêmicos. Ele vai citando durante o livro a íntima relação dele, de como na trajetória profissional dele a academia foi realmente tão importante. E ele cita isso desde do antecessor dele, o professor Edmundo Vasconcelos, que eu não tive a oportunidade de conhecer, seguramente o Kalil conheceu, e depois ele vai citando todos os acadêmicos e como a academia o ajudou para que ele pudesse realizar muito dessa. Então, todos nós acadêmicos ficamos realmente muito partícipes daquela história que é realmente admirável. E, além de ser muito curiosa e muito engraçada, ele vai relatando muitas histórias que eu já conhecia, e o Fábio Jatelli também contou outras que eu nem conhecia, do tiro que ele levou. Então, é muito interessante, realmente eu recomendo. E eu até citei a ele, falei, poxa, o senhor tinha que ir lá no Rio, levar e para lançar o livro. Aí ele disse que ele está com muita dificuldade de viajar, mas eu sugeriria, presidente, que arrumasse um jeito de trazer esses livros totalmente autografados para distribuir a todos os acadêmicos. Eu acho que é um grande serviço que a academia pode fazer, distribuir esse livro aos que não puderam ir por alguma razão. E a segunda coisa, presidente, rapidamente, é só para enfatizar das nossas reuniões em Paris, junto da Academia Nacional de Medicina do Brasil, que nós vamos ter 3 dias lá. Na segunda-feira, no dia 13, nós vamos ter uma recepção que esse ano é o ano do Brasil na França. Então, a Embaixada Brasileira incluiu essa visita no calendário anual das atividades Brasil-França, e então nós vamos ter uma recepção na Embaixada no dia 13 de outubro, às 16 horas. Nós vamos ter, no dia 14, a reunião com a Academia de Medicina da França, que o acadêmico Francisco Sampaio está à frente, está cuidando. E no dia 15 nós vamos ter uma reunião, aproveitamos essa oportunidade, a Academia Nacional de Cirurgia da França vai nos receber no dia 15, onde nós vamos ter 2 acadêmicos, o acadêmico Fernando Vaz e o acadêmico André Berger, vão fazer conferências, e além de outras conferências que vão ser seguidas de um jantar oferecido pela Academia Nacional de Cirurgia aos acadêmicos da Academia Brasileira Nacional de Medicina. Então, presidente, eu acho que é importante isso, nós darmos a divulgação devida a isso, mas nós já estamos com 19 pessoas confirmadas, acadêmicos e alguns convidados que vão estar conosco, e se mais que quiserem vir, serão muito bem-vindos. Muito obrigado, presidente.
Mais alguém? Bom, neste caso, eu passo a palavra ao acadêmico Jorge Kalil para continuar o simpósio. Por favor.
Muito obrigado, senhor presidente. Bom, nós vamos dar continuidade agora, temos mais 3 conferências. A primeira delas, ela também foi gravada, mas a conferencista, se tudo der certo, estará lá para responder as perguntas no final. E a outra conferência vai ser ao vivo. Espero que dê tudo certo. Vocês estão conseguindo me ouvir aí? Prestar mais atenção. So, in continuation to our symposium, I'd like to invite Dr. Ivona Aksentijevich from the National Institutes of Health, National Human Genome Research Institute at Bethesda, Maryland, United States, to talk on the autoinflammatory disease from genotype to phenotypes. Dr. Ivona.
Good afternoon to everyone in Brazil. First, I would like to thank my colleague and friend, Dr. Leo Mendoza, for inviting me to participate in this meeting. As you probably know, I'm attending another meeting in Ireland, and that's why this presentation is pre-recorded. I will try at the end of this talk to connect via Zoom with you to answer any questions that you may have after this presentation. So here is the title of my talk. So what I'm supposed to do for you today is to talk or discuss genotype-phenotype correlations in patients with monogenic autoinflammatory diseases. But what you can notice also in this title is that I added a couple more words which are not written in the program of the meeting. And the reason why I did that was that I would like to convey a message to you about the importance of genotype for selecting or choosing targeted therapies for these patients that sometimes present with devastating inflammatory diseases. You had already heard about the history of autoinflammation, so I'm not going to dwell about that. Except to say that there are over 60 genes that are linked to monogenic assays and probably more than 70 diseases. And the reason for that discrepancy is that pathogenic variants in one gene can lead to very distinct and different phenotypes. So essentially, systemic autoinflammatory diseases are either inherited or acquired diseases of innate immunity. I'm using here the word acquired in reference to the contribution of somatic mutations to the pathogenesis of autoinflammation. So these diseases result from antigen-independent hyperactivation of various innate immune pathways, which essentially means that inflammation is predominantly mediated by myeloid cells, which are shown here. Given that some of these diseases are very severe and lifelong, all of them except for diseases which are caused by somatic mutations are lifelong diseases, it's not surprising also to see that in addition to dysregulation of myeloid cells, some of these diseases present also with dysregulation of adaptive immune cells, particularly B cells, so that many of these patients present with a continuum of immune dysregulation that in addition to autoinflammation also includes features of autoimmunity, immunodeficiency, and even atopy. Some of these diseases have been also linked with predisposition to hematological malignancies, and for all these reasons, molecular diagnosis has become essential for all patients diagnosed with primary immunodeficiencies. So let's start with the discussion of the complexity of genetic heritability of SAIDs. So in addition to these classical monogenic diseases that can be either autosomal recessive, as is the case with some enzyme deficiencies, or cytokine inhibitor deficiency, there are also autosomal dominant inherited diseases or patients that have de novo mutations. Patients that have de novo mutations typically present with early onset symptoms and very severe phenotypes, as is the case, for example, in patients that have inflammasomopathies. There are few diseases which are also linked to X chromosome, and I will discuss one such disease for you, namely VEXAS. There is also evidence for digenic inheritance in patients that have so-called proteasome deficiencies. PRAS or CANDLE are the other acronyms for this disease. But essentially, these patients have pathogenic variants in two different subunits or two proteins that make this complex, protein complex, which is known as immunoproteasome. There are also diseases that are thought to result from oligogenic inheritance of low-frequency, low-impact alleles. Low-frequency alleles are alleles that are typically found in public databases with a frequency of 1 to 5%. Often they are not reported following genetic testing, or if they are reported, they are typically reported as variants of unknown clinical significance. Nevertheless, Crohn's disease is exemplary disease which results from co-inheritance of low-frequency genetic variants in the NOT2 gene. There is also polygenic inheritance in autoinflammatory diseases. Here I refer to co-inheritance of common variants, also known as SNPs. Here we are talking about allele frequency anywhere from 10% to 40 or even 50%, and evidence for polygenic inheritance exists in diseases such as PFAPA or adult-onset Behçet's disease. And finally, in the past 5 or 10 years, there has been increasing evidence for the contribution of somatic mutations to the pathogenesis of autoinflammation. So let's start with monogenic germline variants. So basically, monogenic SAIDs can be classified as IL-1 mediated, NF-kappa B mediated, or type 1 or even type 2 interferon mediated diseases based on their molecular pathogenesis and responsiveness to targeted therapies. However, this classification is more conceptual for the reason that these pathways are interacting and they are interregulated. So it is not surprising that, for example, patients that we think have classical NF-kappa B mediated disease based on the high levels of TNF cytokine in their serum samples or responsiveness to TNF inhibitors, as is the case, for example, with patients that have deficiency of ADA2, also known as DADA2. There is also evidence for type 1 or even type 2 interferon gene expression signature. Here I'm showing you the list of IL-1 activation disorders. Typically, these patients have high levels or they produce a lot of IL-1 cytokine following stimulation of their peripheral blood mononuclear cells with LPS. Testing for IL-1 levels in serum samples is unreliable because the levels are very low. Nevertheless, patients that have severe IL-1 beta activation disorders, they will have high levels of IL-18 cytokines. So these patients present with strong IL-1 signature, and for that reason they have good response to treatment with various IL-1 inhibitors. Patients that have more severe phenotype may require hematopoietic stem cell transplantation, and certainly that's the case with patients that have so-called inflammatory actinopathies. So IL-1 beta activation disorders can result from mutations in different genes. They can result from gain-of-function mutations in cytosolic sensors, inflammasomes, which are shown here. They can also result from biallelic loss-of-function mutations in genes that regulate inflammasome signaling, as is the case, for example, with patients that have mevalonate kinase deficiency. So mevalonate kinase pathway is critical for the production of not only cholesterol, but also certain proteins which are known as isoprenoids, and these isoprenoid proteins are known to regulate, for example, the function of the pyrine inflammasome. These diseases can also result from biallelic loss-of-function mutations in receptor antagonist genes, as is the case with patients that have DIRA and DITRA. Essentially, these patients have deficiency of the endogenous IL-1 receptor antagonist protein or IL-36 receptor antagonist protein. And finally, these diseases can result from mutations in genes or proteins that regulate cytoskeletal signaling, as shown here. In the interest of time, I will just briefly review for you mechanism of disease inflammasomopathies. Essentially, these diseases result from gain-of-function missense mutations in proteins that nucleates different inflammasomes. These mutations could be either monoallelic or biallelic, depending on their location. So mutations that affect this NAC domain, typically one variant is enough to trigger the activation of this protein complex, which is known as inflammasome. In most of these inflammasome-mediated diseases, there is a formation of decamer complex, which can basically involve 10 subunits of the NLRP3 or NLRC4 proteins. Activation of this inflammasome complex will lead to the activation of caspase-1, and then caspase-1 will cleave pro-IL-1 and pro-IL-18 cytokines into its active form, and it also trigger cell death by pyroptosis. Clinical presentation in these diseases is highly dependent on the cell-specific expression of these proteins. So patients that have gain-of-function mutations in the NLRP3 protein, which is highly expressed in leukocytes, will present with severe systemic inflammation, with skin rashes that you will see later on, and sometimes with CNS inflammation, while patients that have gain-of-function mutations in NLRC4, which is highly expressed in the epithelial cells of guts, will present with severe GI inflammation. So skin inflammation is a feature of many IL-1 activation disorders. Essentially, on biopsy, these patients are diagnosed with neutrophilic infiltrates in their skin, despite the fact that sometimes they can present with urticaria-like rash, as is the case with patients that have, for example, cryopyrinopathies or gain-of-function mutations in the NLRP3. So they don't have active mast cells, as you would expect based on this clinical presentation. Rather, they have a lot of neutrophils. The rash could be anywhere from this localized tender and warm rash, which is seen in patients that have familial Mediterranean fever, which is still the most common autoinflammatory disease, to cystic acne, pyoderma gangrenosum type of rash in patients that have mutation in the same protein, pyrine protein, just in a different domain of this protein, or pyoderma gangrenosum type of skin rash, which is seen in patients that have PAPA disease, which is caused by mutations in the protein PST-PIP1, and this protein is known to interact with the pyrine protein. And here you can see pustular psoriasis type of rash in patients that have deficiency of these receptor antagonist proteins, namely DIRA and DITRA. I'm showing you here a few photos demonstrating the efficacy of the treatment with IL-1 inhibitors. On the left side of this slide, you can see how treatment with anakinra, I believe in that time, although at this time I think patients are mostly treated with canakinumab, how treatment with these inhibitors was very efficient in suppressing urticaria, skin rash, and also CNS inflammation, which is the feature of patients with severe cryopyrinopathies, as shown here in these images. And on the right side of this slide, you can see again how therapy with recombinant IL-1 receptor antagonist protein, which is missing, endogenous protein is missing in patients with DIRA disease, is very efficacious in suppressing this skin inflammation or pustular inflammation in this young boy. And this was something that was observed even 3 days after this therapy was initiated. Again, you have to understand understand that these are lifelong diseases, and these patients will have to stay on this therapy for the rest of their life. And if they don't receive this therapy, they are going to flare with systemic inflammation or skin inflammation. NF-κB mediated diseases are, as it says, predominantly mediated by NF-κB pathway, MAP kinase pathway, although these patients, again, if they have severe features or severe phenotypes, they can present with type 1 or type 2 interferon signature. And again, these patients respond well to treatment with TNF inhibitors, or they may require bone marrow transplantation if they have severe phenotypes. NF-κB mediated autoinflammatory diseases are disorders of dysregulated ubiquitination and dysregulation in cell death. So I apologize for this busy slide. However, I can't really explain in any other way the ubiquitination related defect causing these diseases. So here shown is essentially the TNF receptor 1 signaling pathway, which is critical for the activation of the canonical NF-κB pathway. So this whole signaling pathway has to be heavily regulated to provide optimal immune or inflammatory responses. And this is happening through a variety of post-translational modifications that include ubiquitination, phosphorylation of different molecules, etc. So the attachment of these K63 ubiquitin chains and MET1, also known as linear ubiquitin chains, to various molecules such as RIPK1 or NEMO will result in the activation of the canonical NF-κB pathway and pro-inflammatory signaling. Nevertheless, this activity has to be terminated at some point, and there are different ways for its termination, and one of these ways is also by removal of the very same ubiquitin chains from their substrate molecules by enzymes which are known as deubiquitinases or DAPs. So, accordingly, patients that lack enzymes such as LUBAC or ZIAP that can't really attach these ubiquitin chains, so let's say to RIPK1 and NEMO, will present with severe immunodeficiency and some features of autoinflammation. On the other hand, patients that lack these ubiquitinases such as A20 and otolin, which are essential negative regulators of inflammation, will present with the constitutive activation of canonical NF-κB pathway and with severe autoinflammatory diseases which are known as hypoinsufficiency of A20 and otolin deficiency. Here I'm showing you a picture of a patient that was early in life, fortunately diagnosed with otolin deficiency. He presented with severe systemic inflammation, GI inflammation, etc., and which required treatment with high dose of steroids. And here you can see the picture of this patient following few years of treatment with an TNF inhibitor. Interferon-mediated disorders are also known as interferonopathy, so these patients, in addition to high expression of type 1 interferon genes, can also have high expression of type 2 interferons, namely interferon gamma. These patients are nowadays treated with either JAK inhibitors or this new drug, anifrolumab, which essentially blocks the interferon receptor 1 molecule, and some of them, they may require also a bone marrow transplantation. There are several mechanisms of the disease which are linked to interferonopathies. Patients that have this Acad-Gutierrez syndrome, which is associated also with neurological impairments. Most of these diseases are caused by defect in enzymes that are important for the nucleic acid degradation, which are shown here. Interferonopathies can also result from monoallelic gain-of-function mutations in molecules such as MDA5 or STING, which work as sensors for nucleic acid. So patients that have gain-of-function mutations in this STING protein present with early onset vasculopathy, which is known as SAVI. In the past couple of years, there has been more and more evidence that interferonopathies can also result from dysregulation in STING protein trafficking, and that's certainly the case with patients that have Coppé syndrome. I already reviewed for you briefly proteasome-associated autoinflammatory syndromes, but basically patients with PRAS, they have defect in the function of immunoproteasome, sometimes even proteasome, which are expressed in all cells, while immunoproteasomes are expressed only in hematopoietic cells. As a result of that, they will accumulate a lot of undegraded proteins, because proteasome complex is important for protein degradation, and this accumulation of unprocessed or undegraded proteins will trigger different immune pathways, including type 1 interferon signaling pathway. And interferonopathies can also result from defect in negative regulation of this pathway, as shown here. So mutations can be inherited either as dominant or recessive, depending on the protein function. Interestingly, there is a marked phenotypic difference that occurs with the same genotype, even among the same family members, which then suggests the contribution of environmental triggers such as infection, vaccination, or the contribution of some modifying genetic factors, whether they are pro-inflammatory or anti-inflammatory or protective genetic factors in disease onset, expression, and progression. Clinical non-penetrance is a common feature of many interferonopathies, and it's estimated to be 30% for patients that have COP-A syndrome. Here I'm showing the major features of interferonopathies. Many of these patients present with chilblain-like skin lesions. They can also present with this annular kind of skin rash, paniculitis, lipodystrophy, and the most severe complication of disease are interstitial lung disease and basal ganglia calcifications, which are associated with neurological impairments. How do you prove that a disease is interferonopathy? One can do it in different ways, either by doing QRT-PCR, which is the least expensive method, just by checking the expression of 6 genes that are biomarkers of type 1 interferon signature, or by doing RNA sequencing or DNA expression, gene expression arrays, as shown here, or by showing increased phosphorylation of STAT1, either by flow cytometry or simply by Western blots. I already mentioned reduced penetrance in interferonopathies. Here I'm showing you patients that have monoallelic gain-of-function mutations in STAT4 protein associated with pansclerotic morphea. There are fortunately only few patients reported in the literatures, and what I would like really to draw your attention to is this part of the figure. So essentially this patient inherited this pathogenic variant from his mother, who was almost asymptomatic, or she was diagnosed late in her life with hypothyroidism. So we still don't understand, the mechanisms why this her son presented with such a severe and potentially lethal phenotype. Fortunately, these patients, or at least a couple of them that are still alive, have been treated successfully with ruxolitinib. I already talked about proteasome-associated diseases. Here I'm just showing you the pictures of a few of these patients that present with lipodystrophy, especially in the upper parts of their bodies, with muscle atrophies, and also they can present with neurological impairments. Disease activity in patients with PRAS or CANDLE and also other interferonopathies can be controlled, to say, with JAK inhibitors. As shown here is another patient with PRAS or CANDLE, and also pictures of patients that had SAVI disease. Again, here we have to understand that these patients are not cured with this therapy. Rather, their disease activity is kind of kept under control. Well, genetics of human immunological diseases is even more complicated. So in regard to patients that have enzyme deficiencies, if these patients have null alleles, again, these are recessively inherited diseases, they will present with severe congenital syndromes, which are shown here, whereas patients that have hypomorphic variants will present with milder forms of the disease. And here I'm showing you again the example of the other two. So patients that have almost absent activity of this enzyme, ADA2, will present with severe early onset phenotype manifesting as pure red cell aplasia or bone marrow failure, while patients that have some residual ADA2 activity, or basically have hypomorphic variants in this gene, will present with inflammatory DADA2 manifesting with vasculitis, and also predisposition or susceptibility to recurrent strokes. So again, the therapy is different. Inflammatory DAD2 phenotype is highly responsive to TNF inhibitors, while patients that have hematological DAD2 or basically bone marrow failure require bone marrow transplantation. Few words about somatic mutations. So these are genetic alterations that occur post-zygotic in somatic cells, whether leukocytes, keratinocytes, etc., and are not passed on the children. Depending on the time of occurrence, if they occur early in embryogenesis or in post-zygotic stage, these patients will more likely present with early onset symptoms. If they happen later in life, they will present with late onset symptoms. Somatic mutations have been reported in several autoinflammatory diseases, typically dominantly inherited diseases. They often arise at residues which are critical for protein function, which essentially means that germline variants are not tolerated. They are incompatible with life, and these somatic mutations can be identified or detected. The best way to detect them is by targeted gene panel sequencing, but also they could be detected by whole exome sequencing or even whole genome sequencing, depending on the number of reads. In patients with S8, these mutations are found predominantly in myeloid cells, and the frequency of mutant allele can be either very low or very high, and it can increase over time, leading to progressively more severe clinical presentation. Few words about VEXAS, which is the most famous autoinflammatory disease caused by somatic mutations. So essentially, these patients have somatic mutations exclusively in their myeloid cells in the gene UBA1, which is X-linked gene. So that's why this disease occurs only in males. Females can have it if they lack the second chromosome, as is the case with patients that have Turner syndrome, for example. Nevertheless, UBA1 is the major enzyme which is required for the initiation of cellular ubiquitination. So patients with VEXAS present with a variety of clinical features, anywhere from neutrophilic dermatosis, relapsing polychondritis, vasculitis, lung disease, or they can also present, or they will present with progressive hematological abnormalities. Sometimes they are diagnosed with MDS, myelodysplastic syndrome, and the mortality is high. Initially, we reported to be 40%. Nowadays, it's lower because these patients are diagnosed earlier in their life. As for the prevalence, there is one study that suggested it could be 1 in 4,000 men over age of 65 years, although that has not been confirmed in other studies using other public databases. Again, as I said, these mutations are not found in T or B cells or fibroblasts, and variant allele fraction, or the fraction or frequency of the mutant allele, it's very high. It's often over 50% in DNA samples that are isolated from peripheral blood leukocytes. Why order genetic testing? Well, one study from Baylor Hopkins Center reported that 55% of 110 patients clinically diagnosed with primary immunodeficiencies were misdiagnosed based on their initial clinical characteristics. So genetic testing is used to confirm a specific diagnosis, to exclude all other conditions with different overlapping symptoms, to inform about targeted therapies, and for genetic counseling in families. And here again, I will use the example of neutrophilic dermatosis that can result from mutations in different genes, and accordingly these patients require different therapies. So in conclusion, S8s are rare diseases, but they have provided important insights in the regulation of innate immune responses. This genotype first approach in studying patients with S8s and other primary immunodeficiencies has shed light on the spectrum of immune dysregulations and phenotypes which are associated with these diseases, and also these discoveries have played a vital role in guiding targeted therapies and offering insights for new drug development. With this, I would like to thank you all for your attention, and I look forward to your questions.
Nós estamos com o problema de conexão com ela também. Não sei se ela conectou. Conectou? Não. Bom, ela não conectou, então acho que a gente vai para a próxima. Alguém tem alguma pergunta que eu gostaria de deixar para ela? Fala, Marcelo. Anota aí, Léo.
Direto a ela?
Pergunta a gente passa para ela, porque ela não está aí.
Então, in English or
Whatever. Português.
I would like her to, first of all, to congratulate her for such an exciting lecture and ask her if she could speculate on the selective pressures on different populations that maintain those, for maintaining those type of genes, or eventually the disappearance of some of those genes. How the selective pressures work in different human populations.
Thank you. Thank you, Marcelo. I think she's not online, but we'll pass the question to her. I think it's a very important question. And I think that we'll have more and more of this rare disease because of the quality of medicine makes these people survive, even though they have so many medical problems, right?
Eu tenho uma pergunta também, em português, porque acho que não tem necessidade de um americano responder. Uma das doenças que frequentemente é associada com isso é a doença de Sjögren, né? A doença de Sjögren pode ser responsável pelas alterações da mucosa bucal da paciente. Ela não mencionou, mas também causa muito problema de olho seco. A gente sabe que alguns pacientes com doença de Sjögren evoluem muito pior com o tratamento convencional que os outros. E algumas pessoas que usavam ainda empiricamente, há mais de 10 anos atrás, o imunologista reumatologista de São Paulo, Morton Schemberg, costumava, por conta própria, sem muita evidência científica, usar muito biológico. E ele, realmente, eu vi alguns casos que, uau, melhorava muito. Bom, em termos práticos, como é que você pode avançar no diagnóstico diferencial dessas entidades, assim, inclusive para saber qual o tratamento melhor? Obrigado. Pergunta fácil.
Léo, depois a gente passa essas perguntas para ela. I think we should go on. Since we have the new speakers should be online. Are you online, Dr. Rosa Bachetta?
Yes, I am.
Thank you.
Okay.
Nice to see you.
Nice to see you.
Okay, so let's introduce you, Dr. Rosa Bachetta. It's NT and Robert M. Bass Faculty Scholar from Stanford Maternal and Child Health Research Institute, Center for Definitive and Curative Medicine, Division of Hematology, Oncology, Stem Cell Transplantation, and Regenerative Medicine, Department of Pediatrics, Stanford School of Medicine, California, United States of America. And she's going to talk on gene therapy to FOXP3 and IPEX syndrome, a model of systemic autoimmunity. Thank you very much for being online.
Thank you all very much for guesting me. And I think, a special thanks go to Dr. Leo Mendoza, not only because he invited me here, but because of his long-lasting collaboration and work together on this disease, which has been really a big pleasure for me. So I'm talking about IPEX syndrome. IPEX stands for immune dysregulation, polyendocrinopathy, enteropathy, and is an X-linked disease, so manifesting only in males and otherwise carried by healthy mothers, and is really a prototype of monogenic autoimmune disease. Typically, and it's due to FOXP3 gene mutation, which is FOXP3 is a transcription factor essential for the development of functional T regulatory cells in the thymus. And therefore, these T regulatory cells are coming out dysfunctional, are not able to suppress the T effector cells, and therefore the T effector cells full of autoimmune reactive cells are attacking all different organs. And amazingly, the disease manifests immediately after birth. Actually, it is also possible to have the disease in utero, so IPEX can be a cause of fetal hydrops. But generally, these patients are, they at birth, they reach birth as normal gestational age, and then they manifest immediately after, within the first months of life or within the first year of life. As you can see here, with very severe onset, the triad of symptoms which are typical are enteropathy, which is manifested in about 90% or 95% of the patients, and half of them also develop type 1 diabetes. And also the skin is pretty much a target of the autoimmunity. But really, every organ can be target of autoimmunity, especially the blood, the kidney, and lately we also have observed neuromuscular problems in these patients with IPEX. So as usual, because the disease has been really misdiagnosed until several years ago, the estimated mortality is still 40% if it's not diagnosed promptly immediately after birth. And overall, considering this patients reported are, about 600 patients have been reported for about 200 different mutations. However, importantly, what has become clear more recent year is that IPEX can also have a little bit of a different clinical presentation. I would say atypical IPEX or more moderate disease. And typically these patients have later onset or less severe, but anyway, chronic manifestation of polyautoimmunity, which, is really a cause of very bad quality of life and always being, healed at some degree. So this is very important, and it is also very important to understand that IPEX, as I said at the beginning, is a prototype of autoimmune monogenic disease and goes really together with many others that have been discovered after, where really the genes that are affected by a mutation are genes encoding for proteins that are essential for the function of T regulatory cells. And when we talk about T regulatory cells here, we talk especially about the ones that are coming out of the thymus, which, in the CD4 positive peripheral blood mononuclear cells, they represent only 4 to 7% of these CD4 positive cells. However, they are very strong regulator and very important for life. And as you can see, there is transcription factor, BAC, Helios, there are signaling molecules, there are receptors like the CD25 receptor for IL-2, which is essential for the survival of T regulatory cells in the periphery and for their function. So by many different hints, these molecules are all important, and they all comprise these pathologies which are called T-regopathies. But, very interestingly, you can see that there is a lot of overlap between T-regopathies, some kind of autoinflammation also in some of the IPEX patients that is coming up, and all form the primary immunoregulatory disorders. Interestingly, it's good to remember that, you know, there are altered Treg number and function also in case of non-monogenic diseases, such as allergy, fetal maternal tolerance, graft-versus-host disease. So T regulatory cells are really essential for a healthy life. Going back to FOXP3 mutation, in the gene, there are different domains which are very important, and interestingly, the patient, you know, they really can have many different mutation sites. So there is the 4CAD domain mutation, which is here. It's yeah, definitely a more dense of mutations, but indeed all the genes can be all the different parts of the gene can be affected by different mutation, which very often are missense mutation, but also sometimes deletion, shift, and so on. So when the mutation of FOXP3 affects the T regulatory cells and the T cells in general, the T cells tends to proliferate more, and the T regulatory cells not to suppress, as it is shown in this case. Interestingly, from the immunological point of view, it's not easy to recognize, you know, at the first blood draw is not so easy to recognize the disease. These patients, you know, in primary immunodeficiency, you don't see the T cells, the B cells, etc. In this case, you see a normal lymphocyte count or even higher, and with higher CD4 than CD8 and normal B cells and NK cells. However, there is almost always eosinophils that are increased, as well as the IgE, as in other similar immunodeficiency with immune dysregulation. There is also the presence of specific autoantibodies, especially in patients that have enteropathy. There are anti-harmonin antibodies that are found only in IPEX and not so much in other diseases with enteropathy, including Crohn or other IPEX-like disease. So anti-enterocyte and called anti-harmonin antibody are very peculiar of IPEX syndrome. But what is very interesting and important from the diagnostic point of view is to understand that the FOXP3 protein most of the time is present and expressed, although it can be a little bit lower than normal, but as you can see, it is pretty much expressed. So to just phenotype the T cells with FOXP3 may not be significant and helping in the diagnosis if the FOXP3 is normally expressed. However, there are some cases in which the FOXP3 is abrogating the protein expression, and in that case, it's pretty clear that the consequence of the mutation. So what we have been studying over the years is this marker, so the epigenetic marker called TSTR demethylation, that can be measured in the peripheral blood. This is a specific site on the DNA next to FOXP3 coding region, which is always demethylated in T regulatory cells only, and therefore can be quantified just by a simple PCR. And so we see in patients with IPEX that even if by flow cytometry, the T regulatory cells seems to be there, although with quite a variability, and the FOXP3 protein is there by flow, but with this qPCR, we can see that all the IPEX patients express very high percentage of cells with the TSDR demethylation. Importantly, we have observed that this is, really also an index of the disease, the different disease, IPEX disease, so it can distinguish typical IPEX from atypical IPEX, where typical IPEX really have this TSDR demethylation much higher than the other one. And it is also very useful to monitor that over the course of the disease. You can see here some measurement of newborn TSDR demethylation, where basically these are normal values at birth. However, in patients in which the disease is going to be manifested very severely, you can see that at 3 weeks of age, already the TSDR demethylation is very, very high and very altered. So we think that this parameter is really very useful to better, to complement the genetic diagnosis and to really understand more in a prognostic way where the disease is going. Other than that, IPEX is really a disease with a very complex pathogenesis. We have said already that there are autoreactive T cells that are expanded, and these autoreactive T cells are also comprising T regulatory cells or T regulatory cells 2B that, however, because they don't have a functional FOXP3, they actually become effector cells and therefore drive the pathogenesis of the disease as well. This is all associated with increased inflammatory cytokines and with the presence of autoantibodies that are also mediating the damage of the organ. So the disease has definitely limited therapeutic options, and over the years, we have seen many progress towards a better treatment. The disease was really discovered at the beginning of 2000 when the gene and the T regulatory cells and the human disease and the mouse model were all, reconnected and understood. And then, over the years, we have contributed in different ways and increasing the understanding of the mechanism, the natural history, and now really in the last 5 to 8 years, we have really focused on looking at gene addition and gene correction as alternative best treatment for these patients. What currently we have available is, of course, immunosuppression. And among all different immunosuppressive approaches that can be taken for IPEX, rapamycin or sirolimus is one of the best accepted nowadays, and patients that tolerate and can be compliant with the administration of sirolimus really see advantage. We have seen that rapamycin can really improve the function of regulatory T cells and their survival, even in the presence of FOXP3 mutation. So this is a very, very good pharmacological approach. Otherwise, of course, allogenic hematopoietic stem cell transplantation has been curative for many patients with IPEX. However, is not always available and still is limited in the donor availability, is quite a heavy interventional treatment that many of these IPEX patients, when they are in poor condition, they cannot even approach the conditioning required by the transplant. And there are, in the outcome of the allogeneic transplantation, there are mixed outcome, not really clear. We don't know exactly if mixed chimerism is sufficient to control the disease. And there are a lot of rejection or failure higher than in other primary immunodeficiency, as well as persistence of autoimmune symptoms. And here you can clearly see that, in the black line, this is a graph of percent disease disease-free survival over 25 years. You see that patient with only immunosuppression, they slowly have, more problem and recurrent symptoms and side effect of the treatment, whereas the percentage disease-free survival after allogenic transplantation is about 60%, which is, good if you're cured, but it's definitely not enough, also considering that the transplant is not for everyone. On the other hand, the disease has many different characteristics to be really a good candidate for gene therapy, and over the years, my group has been really worked on, sorry, I have a problem. Yeah, really worked on different gene therapy approaches. So for gene therapy, the first approach that we have pursued is the one on autologous T cells, so CD4 positive T cells. So in terms of generating, with the aim of generating a Treg replacement therapy, so giving functional T regulatory cells to patients that don't have them. And why we do that in a way that we basically collect the CD4 positive of these patients from the blood. We purify them and we convert them into T regulatory cells by transducing them with a construct which contains FOXP3 wild type under a constitutive promoter, so that the FOXP3 gene is always expressed at very high level, as well as the naturally occurring T regulatory cells do. And together with the FOXP3, we also have in this construct a surface marker, which is expressed by all the transduced cells. And this is very useful because it allows us to purify the cells during the manufacturing, but also to monitor the cells that we infuse into the patient. So this is basically, as I said, a Treg replacement therapy, and we have opened the trial at Stanford. And it is now open, a phase 1 study, which is also given the designation as an orphan drug designation to this cell product. So the cell product, as I said, is really identical functionally to the T regulatory cells that are coming out of the thymus. In the main marker, they express regulatory markers which are essential for their function in the red line column, and also they are hyperproliferative and they have very highly suppressive function. We have done many experiments, of course, in the preclinical stage of this trial, showing that these cells are very potent in suppressing, in humanized mice, in suppressing xeno-GvHD, or also a model of humanized IPEX disease, whereas they don't impair the ability of the T effector cells to fight pathogens. So the trial that we have open is open now at Stanford for patient age up to 35 years of age with a detected FOXP3 mutation in the presence, of course, of clinical history of IPEX with or without ongoing immunosuppression, and which are also patients that are refractory to treatment or had unsuccessful stem cell transplantation before. The design of this trial is described here. we are testing 3 different doses of the cell product, 1 million, 3 million, or 10 million in a single patient. And we have 2 arms. One arm, the one that we call cohort A, is the one with patients 12 years of age or above. And the cohort B is the one with patients below 12. And 1, 2, and 3 are the 3 different doses. And really, this is a phase 1 study. So the main endpoint are safety and feasibility of making this cell product from different patients with different mutation. And also, of course, follow the impact of the disease manifestation and gain information on the pharmacokinetic of in vivo survival and stability of this Treg-like product and its effect in patients with, an altered immune system. So these are in yellow, the patient that we have already treated. Basically, we have enrolled 11 participants and we have treated 8. 2 are waiting for the infusion. Several of them are out of 1 year study, and which is, 1 year is the most intense follow-up, although because they have received gene therapy, we actually have to be in contact with the patient for basically almost 15 years. These are the different characteristics of the patient. The age is from 26 to 9 years of age. They have different mutation and they really resemble all the different clinical manifestations that I already described to you. Most of them are under silorimod treatment. This is the cell product, which is highly pure for FOXP3 and NGFR, which is the marker gene, and with very good suppressive activity and vector copy number. The cells, interestingly, after the manipulation, have a high percentage of stem cell memory, which is a very promising feature that suggests that the cells are prone to survive in vivo. And all the different product that you can see here for 9 patients shown here, 8 patients, are very reproducible and really very similar to each other, as shown here, analyzed at the single cell level. When we look in the blood of the patient, we actually see that the transduced cells, which are the one that I show you here, are the FOXP3 positive and CD271 or NGFR positive. They really are comprised within the CD4 positive CD3 and within also the CD25 high and 127 low, which is where we should expect to see the T regulatory cells in the human blood. And at the single cell level, again, we can see the signature of these cells in the blood of the patient. They express very high FOXP3. They express Treg marker, although they also still have their feature of T helper cells. And here you can see they all express very high level of FOXP3. But they don't produce the cytokine for example, Th2 or IL-13 or Th17 because they are inhibited by the strong FOXP3 expression. So basically, overall, for this initial trial, we know that we maintain the parental CD4 positive T cell heterogeneity and also the TCR specificity, but acquire Treg signature in this CD4 LV FOXP3, that they are enriched of stem cell memory cells and they can be traceable in the blood, and so far they are also showing functional stability. In the IPEX patient, we can conclude that is feasible, this treatment. The infusion has been safe in all the patients treated. We never had any problem, and we also have promising evidence of beneficial impact on the disease manifestation, although we could not yet suspend the immunosuppression. So that's, I think, would be for a phase 2 trial. So we hope that this is really going to expand as a subsequent trial, and also in different application, not only in IPEX, but in other autoimmune disease. I just want to mention that, of course, you know, over the years, we have been collecting so many patients. IPEX is distributed really all over the world. And collaboration, international collaboration, have been really fundamental to gather all the information I told you. Without international collaboration, all this would not have been possible. And also, I would like to leave you with what is ongoing in my lab, which is looking for gene correction in hematopoietic stem cells using CRISPR-Cas9 to really reestablish a normal lymphocyte subset, which can guarantee a long-lasting treatment of these patients, always using autologous blood stem cells, and can be really resembling all the feature of T regulatory cells, and this is in preclinical development. I really need to thank all, you know, a lot of people participating to this study in my lab, but especially in the clinical trial office, the clinical team. Dr. Alexander has been really fantastic in following these patients. A lot of agency that gave us funding, in particular the California Institute for Regenerative Medicine, as well as the FDA. And also the patients, the different, people in my lab, international collaborators, Dr. Leo that I already mentioned, and most of all, always, all patients and the family. And thank you very much for your attention.
Thank you. Thank you very much, Dr. Bachetta. Since you are online, is there any questions from the floor? Well, I can start. Well, we are pretty sure that it's very difficult to keep these regulatory phenotype, not only in those IPEX, but in everyone. I mean, this is the dream of all the people that work on autoimmunity, to have T regulatory cells that you could inject and keep them going. So the question would be, how many cells should we inject? Are you going to select cells that have a specificity, since this can change? I mean, of course, you're using a lentivirus to keep the profile of regulatory cells. But do you think that in the environment, the body environment, these cells will be kept this way, well, or they are going to disappear or be enhanced too much in the too much volume, that it became dangerous in the regulation of the immune system? How you can regulate the system, by the way, since you are injecting a cell with a fixed phenotype, that the fixed phenotype was done by injecting a lentivirus?
Yeah, all extremely good question. So what we are learning from this trial, we reach the maximum, so the highest dose is 10 million per kilo, which, you know, it's already quite an impressive number of cells that we are giving to the cells, to the patient, if you consider a 40 kilo or a 60 kilo, or, you know, even less, but it's really already something. And we clearly see a dose response. However, we, interestingly, going from 1 million to 3 million, we see a 10-fold increase in the number of cells that we see in the blood. But going from 3 million to 10 million, so we don't see as much increase. So this suggests that, you know, there is probably 10 million is already a good number. But what is different is the persistence of the cells. So giving 10 millions allows the cells to stay there for longer time in the peripheral blood. So what I think, we are contemplating is to give more than one infusion over some time, like over the first 2 or 3 months, and that would help in reaching, a more stable amount of cells. Of course, the long term, it's a problem, but we don't know if they can stay forever there. We know that for sure when you have good regulatory T cells, they are contagious in their tolerance. So they exert what's so-called infections tolerance because they create a tolerant environment. And this could be very beneficial for IPEX patients which have, a very acute disease and that cannot even be controlled by more than one pharmacological treatment. Also, other things that you have touched are the other question is the antigen specificity. So what we know that we start from in the IPEX patient from a high frequency of autoreactive T cells, and we know that we can preserve this autoreactivity. So we make actually a lot of Treg or Treg-like that are autoreactive in their TCR, but now they are regulatory. So this is very good. This is also good for IPEX that have, many antigens because they go against many different organs, and may not be the case when you only have one disease targeted by the autoimmunity. In that case, may be more useful to have an antigen-specific Treg. But yeah, so then a different approach could be thought. Mm-hmm.
Yeah, I think that's a very interesting point. That you do not discriminate the T cells that will be regulatory because IPEX you have so many antigens,
right?
And you have to be regulatory for everything.
Mm-hmm.
But do you think that if it works well, we could think in the future to have antigen-specific regulatory cells to be injected to cure a specific disease?
Yes,
like multiple sclerosis.
Absolutely. Mm-hmm. Yes, okay. I think that would be ideal. Absolutely, yes. Mm-hmm. So I see, we can learn a lot from this trial, and a lot of information can be instrumental for the next steps of this kind of treatment.
I wish you a very, very good luck because it's not a simple. It's not a simple clinical trial for sure.
Thank you.
Any other questions for Dr. Rosa Bachetta? No? Well, so thank you very much from from the National Academy of Medicine from Brazil. It's a very interesting and very promising, I would say, approach, and this is ideal to have a regulatory cell to be injected and keep this function for a long time. I hope it'll be successful, and we can take advantage of your studies to treat many diseases. Thank you very much.
Thank you. Thank you very much.
Well, I think now we should continue on. And now we have Dr. Leonardo Oliveira Mendoza that works in my medical service at the School of Medicine, University of São Paulo. And he's going to talk on autoinflammatory diseases in Brazil, challenges and opportunities with the construction of a national center for inborn errors of immunity and immune dysregulation. I don't know if Dr. Bachetta is still with us or she left.
I'm still here.
Okay, so you're going
I'm going to speak in English, Professor.
So now
Oh,
in English.
it's up to you, but no worries if you want.
No.
Okay, no worries.
Keep in English.
I was
It's good
programmed
training.
to speak in English. So I would like to thank for the invitation here today to talk about the project I'm conducting for the last year. That is the construction of the National Center for Inborn Errors of Immunity and Immunity Regulation. And of course, I could not do this alone, especially with the help and assistance of Professor Jorge Kalil and Professor Magda, but all with the international friends that you could see here today, Professor Rosa Bachetta, Professor Gattorno, and Ivona. So I'm not going to spend that much time on talking about the immune system, but as we can see, the huge number of genes discovered over the last years associated with the monogenic forms of inborn errors of immunity, we can see now that in the USA, the estimated prevalence of all inborn errors of immunity to be of 6 in 10,000 individuals, which is not more a rare disease. So we really have to think about these disorders in our daily practice. And when we come to the point when we see the data of the registered case of inborn errors in South America, we see a small number of patients reported in the last report of the Latin American Society for Immune Deficiencies, with only 2,000 patients reported, which accounts for 1 in every 100,000 individuals, which means that we still have a lot to work on these situations here in Brazil to bring the diagnosis and the quality of life that we want to these individuals here in Brazil. And that's why the professors in the USP University in the School of Medicine, they proposed the creation of a national center for inborn errors of immunity and immune regulation to come across the individuals that are not yet diagnosed all over Brazil. So, sorry. So, as Ivona said, and also what Professor Kalil said, we have to always remember that inborn errors of immunity is not just a disease from the pediatric scenario. So as we can see here, we all know about the severe combined immunodeficiency, but we have many disorders reported only and exclusively in adults patients. As we can see this picture of a patient of mine from the Center of Rare and Immunological Disorders at the Hospital of the Jules that just manifested this disease in the last 6 months. So we really have to think about autoinflammatories and other inborn errors of immunity also in the geriatrics, because these patients also, the disease may also affect adult individuals. And especially when we talk about the VEXA syndrome, that the most famous somatic autoinflammatory disease, it may be present in 1 every 4,000 male individuals, adult male individuals. So it's really, really common disease, and we may have been skipping or not diagnosing the VEXA syndrome in our current in daily practice. What's much more interesting is because, especially for a SCID patient or severe combined immunodeficiency, we already have a target therapy or the best therapy, that is the bone marrow transplantation, and there is a huge number of gene therapies ongoing in the last years. But when we talk about adult onset disease, especially the VEXA syndrome that starts over the 60s, bone marrow transplantation is not yet a possibility. So we have to really think about other therapies for adult individuals as they come to our department in these situations and very severely affected disease. So we really have to think about this disease that also affecting adults and also target therapies for this population. So why we need, what we need to the diagnosis of inborn errors, as we also heard about the inborn errors of immunity in the last topics, it is considered a rare disease, but when you come to the epidemiology of inborn errors in the United States, we see that it's not that much rare anymore. So we really have to think about these disorders in our departments. As we saw the many lectures before, it is a complex immunogenetic interaction that comes to the clinical manifestation of these individuals. So we really need to have training, medical training in immunology and genetics to the recognition and both the diagnosis. And it's essential to have the opportunity to have the genetic sequencing to the diagnosis of these patients and also do immunophenotyping. But it's also important to recognize that not all the genetic sequencing are the same for the same patients. I can clearly demonstrate here, for an example, this child can be clearly diagnosed with a simple next generation sequencing because of the depth of the sequencing that is just very superficial, because all the cells here must be or may be mutated with the gene. But when we come to this adult department, when we come with this adult case, for an example, only a few cells of this patient will be mutated. So we need a different type of sequencing in this situation, especially for somatic mosaicism, in which we need for at least 1,500 in depth of sequencing to find the mutation of the species. So we really need also personalized diagnosis for each situation, and it's very important to have a center for this. And that's why Professor Magda, that is the head of this project, proposed the creation of the National Center of Inborn Errors of Immunity and Immune Dysregulation. This is a project proposed by Professor Magda Carneiro that is funded by FINEP, but we have in the councils of directors, Professor Jorge Kalil, which is also a principal investigator, and Professor Esther Sabino, that is also a principal investigator in the laboratory. The project that I'm going to talk was funded last year, and that we started to work to create this national unit for diagnosis of these disorders in the School of Medicine. So how does the project, what is the proposal of the project? The project is proposed to sequence at least 3,000 individuals in Brazil with suspected inborn errors of immunity independently of the age of the disease or the disease onset. So we are going to sequence it from childhood to geriatrics to all individuals that come to the departments of our collaborators. We are also going to do a biorepositorium with DNA, with cells, serum, and RNA of these individuals. We are going to use the network established by the Brazilian Consortium for Inborn Errors of Immunity and also for the Brazilian network for autoinflammatory disease and immune dysregulation disorders that Professor Kalil and I formed in collaboration with France. And we are going to work with all states of Brazil. So we have collaborators and friends in several public institutions of Brazil that are going to collect samples and collect clinical data of these patients and send to us in the laboratory, in our laboratory in the University of São Paulo, to genetic sequencing and also to genetic validation of new variants, new genes, or new causative mutations in these individuals. So for the clinical data, which is very important to have the clinical data, we gathered information from Professor Gattorno, as he is the president of the Eurofever registry for autoinflammatory disease, and in collaboration with France, we also collected clinical data, how they do the clinical collection data in France, in Paris, actually, in France, but coordinated by Professor Sophie Georgine in Paris and Marie-Louise also in Paris. So we decided to use REDCap, and we have almost 400 informations that is collected in the REDCap. So I'm not going to talk about all of them, but here is just to have an information of all the clinical manifestations that these patients have, and especially, not especially, but one of the most interesting in what triggers the disease, so identify what we can do also for clinical prevention. Our idea is to follow up these patients for at least 10 years, and yearly we want to have an update on the clinical manifestation, on outcomes, and also in vaccination schedules that we are collecting here. Also, another thing that we are working on this project is to disease awareness and continuous medical education. So I proposed the Jornada Itinerante do COBEI. It's a symposium to talk about all the inborn errors of immunity in childhood and also in adulthood. So the idea is to go to the place of our collaborator, to invite the clinicians, all the neonatologists, or the rheumatologists, pneumologists, or all the clinicians that work in that site to talk together about clinical scenarios, or moreover, hear about how do we do genetic analysis, because it's very important for these physicians to understand what is necessary to do genetic analysis, how does it take so long sometimes to have the report. So this is what we are doing since the last month, and we are going to do until the next year. We are going to travel from almost all over states of Brazil to have this medical awareness. We have also some collaborators, some financial support from the Grifols, Octapharma, and AstraZeneca that are working with us to financial support this project. Finally, what we all wanted is the project supported the acquisition of this very modern next generation sequencing machine from Illumina, that is the NovaSeq machine, which here you have the picture of the machine with Professor Magda Carneiro. It was bought on the middle of the last semester, so we are going to start using from the last month. So we are going to have we already have some results, but we are going to have some more robust results in the following months. And we also bought the Imagine, that is a very modern platform that uses artificial intelligence to genetic analysis, which is very interesting because I was from the time when we were doing this by hand, and now we use artificial intelligence that takes just 1 hour sometimes to do the genetic analysis. That's very, very a brilliant acquisition for the project. We already had the Illumina training day. This is the laboratory of Professor Kalil, in which we had the training for the use of the platform. And this is the workflow that we are going to use for the genetic validation of non-synonym mutations. I'm not going to go in depth with this, but if someone wants to know our flow chart, I'm more than able to share this. So this is how we are going to do. I'm going to bring some preliminary results. Up to the last week, we already have 1,224 individuals, samples received in our laboratory, mostly from the southeast and the southeast and south of Brazil. So in majority of them from the state of São Paulo. But we have 53 centers already approved, and we are going to have probably in the next year much more samples from the north region of Brazil, which we don't have that much sample already received, which is a brilliant result in just some months of the project to have that much samples received. We already have the results of the sequencing of 224 individuals, in which 159 of them are negative and 10 were positive. So 4% of positivity for this first analysis, mostly of immune dysregulation because of the characteristic of our the project, how the project was initiated. We also had the chance to reanalyze 70 genome plus exome sequence that were done in the past that were all negative and with the use of the artificial intelligence, we already have some variants that can be validated or considered at least for the diagnosis in these individuals. So it's very important to recognize that it's necessary not just sequence, but also to reanalyze these individuals over the time to have much more diagnosis. Moreover, and finally, we have already 10 students from the graduation that are doing their thesis in the inborn errors of immunity using the data gathered from this project. We have 4 students of internal medicine department that are doing their thesis also with inborn errors of immunity. We have 2 PhD students working on the project and 6 yet to come to preparation to be admitted to the PhD program. We have 2 postdoc working on this project. One is me, that is working very hard. And moreover, and finally, actually, I think that the most important result is we have been diagnosed, that have been explaining the disease that they have in Brazil. So that's the most important. Coming to the top with the challenge and opportunities of the creation of the centers, I point out here some challenges that we already have. So the sample transportation from some states far away from São Paulo, it's really challenging, especially from states in the north, because the sample to come to São Paulo, it's the time to come to São Paulo, it's sometimes complex. Adherence to data collection, so physicians to get very use it to fulfill the data from the red cap. It's been challenging for us too. And also I would like to put here some challenges here, that is the time for DNA extraction, sample sequencing, and the time for the genetic report. So we have to work more on that. But we have many opportunities with this project, because Brazilian population is unique in time of genetics, and it's a piece that was missing in the genetic puzzle in the world. So we really have like a very interesting data of genetic immunological disorders. So it's really a great opportunity for collaboration, also for discoveries, because the probability of finding new genes, finding new pathways, discovering new opportunities for drug development with this project, it's something that may happen in the next years. And finally, this project will allow the generation of robust data that can guide political policies in inborn errors of immunity in Brazil and some other rare diseases. So I have here some 6 to 7 challenges and opportunities that I can find in this project, but we can have so many more. Finally, I would like to thank or obrigado or grazie mille, I don't know which language, but I would like to thank for all the people in our department, the residents and the colleagues that have been working also for this project. And here you have a picture of our big boss and our colleagues, the people that are working in my project. So if anyone has any question also, I'm more than happy to talk or to reply, or if you want to collaborate also, I'm more than happy to include in our project. I hope I have finished in time for...
Okay. Dr. Bachetta, do you have any questions?
No, I was just congratulating with, Dr. Leo. I mean, I think it's amazing. You are a driving force, and Magda, you should be so proud of all this very good work, and count on me for any possible help. Mm-hmm.
Thank you very much, Prof.
Thank you
Also,
very
if
much.
you want
Ivona
to set
Ivona is online now.
I think is mute, maybe.
She's muted. Aksentijevich.
Do you hear me now?
Yes, I can hear you.
Okay, okay. Good evening to everyone, and I apologize for, joining this meeting late. I think I had miscalculated the time difference. I'm in Ireland now, and I thought that there is 6 hours difference between Ireland and Brazil. And for that reason, given the program that my talk was supposed to be at 6:45 p.m., I thought that, I should join you after midnight. So, but I still don't know what is the time difference, between us now. But nevertheless, I'm happy that I had a chance to hear both presentations, and I congratulate to both of you on really wonderful talks. So, Rosa, I don't really have any question for you. I was, just thinking like, why you haven't done your gene therapy in CD34 positive cells. I know the reason for, going straight into T cells, but I was just wondering when listening to your talk, what would be the case if you did gene therapy, in hematopoietic stem cells? So, probably would take a little bit longer time for cells to differentiate into the proper cell type.
Yeah, thank you for the question. And so I think, this particular construct with lentiviral vector and a constitutive promoter, not an endogenous promoter, is not suitable to go into CD34 because we actually have a paper about that. It really gives too much of the FOXP3, so all the immune system is shut down because of too much expression. So the transcription factor FOXP3 has a very tight regulation. And you can imagine that is highly expressed in very few cells all the time, but is transiently expressed in other T cells only upon activation, and then it goes away. So this tight regulation requires really to go with endogenous promoter. So the way we see that is to intervene with a CRISPR-Cas9 approach in CD34 positive, and this is what we are doing. And then replacing the old gene after all the regulatory region and the promoter region, which remain endogenous.
Okay, thank you very much.
Professor, I have a question.
Yes.
Which organ was the most repopulated by FOXP3 in the end in all the patients?
You mean in the trial?
Yeah, in the trial. After the trial, which organ?
Well, we cannot test all the organ. We just had the opportunity to study a few GI biopsy. There is also the problem that these are for patients that are below 12, we cannot do any biopsy. So we can only have some biopsy done in the more adult one. And we see that in the GI, we see that these cells are there. They cannot really be quantified because it's difficult, but they are there. So, and we also know at the single cell level that they express a homing receptor for skin and gut, etc. So, which is a good requirement to suggesting that they go into the tissue. What we have introduced is the possibility to do bone marrow aspirate. In general, in our body, what the Treg that are in the blood represent only 2% of the whole Treg pool that we have, and whereas in the bone marrow, 20% of the Treg are present. So that's what we are going to do next.
Great, amazing.
So, Dr. Ivona.
Yes.
So there are 4 hours difference between here and Ireland. Just
Wow.
to know.
Thank you. I thought
we are 4 hours ahead. You are 4 hours ahead. So there was a question from the floor asking, how do you think that these genes are kept? What about the negative selection of these people? Why those genes do not disappear, since you have those different diseases and people may die of disease? How these genes are kept in the population and do not disappear?
Yeah, I think most of the mutations are actually selected
under positive evolutionary selection because they provide better immune responses, better inflammatory responses. And that's certainly something that we showed, for FMF-associated mutations. So familial Mediterranean fever is still the most prevalent monogenic autoinflammatory disease, very common in Middle Eastern population. And study from our lab showed that individuals who are carriers for these pathogenic variants, actually they produce more IL-1 beta cytokine when their PBMCs are stimulated, which provided them better immune responses against endemic infections. And our study actually showed that this being carrier for FMF-associated mutation is beneficial at a time when plague was common, in Europe and Middle East and so forth. So that's why we are thinking that these variants have been under positive evolutionary selection in people who are carriers, right? Individuals who have two of these mutations, or, if this is some, let's say, dominantly inherited disease, of course, they're going to disappear. But being carrier for some of these recessively inherited mutations probably has been beneficial. And it's been known now for over 20 years that individuals against who are carriers for FMF-associated mutations, they have elevated acute phase reactants. That has been known now for many, many years. It's just that they are not symptomatic. So.
Yeah, that's very interesting. I wonder whether this could be applicable to carrier of FOXP3 mutation as well.
Could be, it could be.
But it's difficult to study that, right?
It is difficult to study. I mean, the way how we did it with FMF carriers is actually that we got PBMCs from asymptomatic carriers. And then we had to stimulate these cells with LPS, and then we measured IL-1 beta production, and we did also animal model studies, which showed the same thing. So that's the difference between inflammation-associated mutations and mutations that are associated with classical immunodeficiencies, because they really don't protect you from. And, I mean, you can think about that, these autoinflammation-associated mutations that have been under positive evolutionary selection, or mutations that, are maybe associated with susceptibility to I mean, they can predispose people to autoimmunity. So that's what I'm trying to say, because they are there in the general population, and maybe it just takes another environmental trigger or something to make these cells even more active. And that's how people, become over the years, the prevalence of autoimmune diseases has increased.
Yeah, interesting. Yeah.
Well, I'd like to, in the name of the National Academy of Medicine for Brazil, I'd like to thank again Dr. Aksentijevich,
yes,
and Dr. Bachetta for being present and giving so nice seminars that illustrated a lot our meeting, and also Dr. Leonardo for bringing new insights of how we're going to deal with this in Brazil. And we are going to stop here, and thank you again. Thank you.
Bye-bye.
Bye-bye. Thank you very much for giving me opportunity to participate in this meeting.
Yep, likewise.
Thank you.
Bye.
I would like to congratulate all the speakers for this symposium. And agora só me resta dizer boa noite para todos, dizer que a semana que vem, infelizmente, eu não estarei aqui, estarei em Coimbra, e a sessão será presidida pelo nosso muito competente primeiro vice-presidente, Antônio Giannina. Sentirei falta da academia. Muito obrigada. Boa noite.
Não.